by students at universities here in Nova Scotia. These are short and sweet presentations, like lightning talk style, so they'll be about 3 minutes each, and we will reserve all questions until the end. So then we'll have a question period at the end of all presentations.
Perfect. Okay. Hello, good evening, everybody. I am Devi. I am a PhD student with the Faculty of Computer Sciences at Dal. I try to use machine learning to estimate marine populations under the supervision of Dr. Christopher Witten. I'm going to give you a short talk about what I do for research. Ocean temperatures, as we all know, have been skyrocketing over the last few years. This graph shows how hot the ocean has been each year. In March 2023, it was the hottest it has ever been, and it has stayed that way since, unfortunately. Look at the orange line for 2023. Now we're in 2024, it's even hotter. See that solid black line there? That's where we are right now. Water can absorb a lot of heat, so much more than metal can. The seemingly small rise of 1 or 2 degrees Celsius means the oceans have absorbed a ton of heat. Because of these higher ocean temperatures, we are seeing stronger storms like we have experienced the last year, coral bleaching in several parts of the world, and disrupted marine ecosystems. For example, about 10 billion snow crab, roughly 90% of their population, have disappeared off the coast of Alaska between 2018 and 2021. Warmer waters have forced these crabs to change what they eat, where they live, which has significantly hurt their numbers. This has put a lot of pressure on Alaskan fishermen, whose industry is worth about $150 million US. Also, we are seeing more tiger sharks now in new places further north than we have ever seen before. This has also been directly linked to warming ocean temperatures. These are just examples of 2 very observable huge changes in the oceans, but there are so many more changes happening that we cannot easily perceive. We need to develop ways to study these changes. Currently, marine populations are monitored by tagging fish, conducting manual field surveys, visual observations. These processes can be invasive, can harm fish sometimes, and do not scale well. How can we dramatically scale up this process? That's where we come in. We are trying to see if we can use cameras to monitor marine populations under the ocean. Covering vast regions of ocean to study marine life is an incredibly challenging task. We have collaborated with DFO, who have deployed several hundred affordable cameras across a large region, specifically in a marine refuge in Newfoundland. Crab pots were fitted with cameras, bait to attract fish, also light sources, because there isn't much light past these depths in the ocean. Fishermen then take these crab pots, drop them in the ocean, and then collect them in their subsequent trips. These videos, these cameras record terabytes of data, which are next to impossible to process manually. We use machine learning to automatically estimate marine populations from this data. So far, we have seen that the machine learning models have been working really well, achieving results similar to manual analyses. We are now working with DFO to significantly scale up this operation by deploying thousands of cameras this season and developing the models to extend to this huge set of data. We are hoping that we will be able to develop technology that will enable us to better understand the state of the ocean and perhaps even uncover exciting marine life behavior probably unknown to mankind. Thank you.
Wow.
Hello, everybody. My name is Emma Jean. I'm a graduate student in biology at Acadia University, and today I'm going to be telling you a little bit about my master's research project, which involves exploring the phylogenetic classification of freshwater mussel, Nephronaias tempisquensis, in Costa Rica. So you might be wondering, why are we so interested in freshwater mussels? And it is because they play an extremely important ecological role in aquatic environments through their filter feeding and water purification activities. Oops. They are also highly vulnerable to, drastic changes in their environment due to their sedentary life history. The larval Glaucidia stage are obligate ectoparasites to fish. And they can also be very successful invasive species in areas that they are not native to. So through my research, I have identified 3 key knowledge gaps that I would like to address, with the first being that there is a pretty noticeable unequal distribution of mussel monitoring efforts in neotropic regions like Costa Rica. Many bivalve genera are pretty understudied, and there is a lack of published molecular sequence data for many taxa, including the genus Nephronaias. So our samples of Nephronaias tempisquensis were collected near an aquaculture farm in Cañas in the Guanacaste province of Costa Rica. And a recent study, a little bit of previous literature, was done by Pfeiffer and colleagues in 2019, where they discovered a new distinct taxonomic clade featuring only Mesoamerican species in the Embleminae freshwater mussel subfamily. And with this, they actually predicted that other genera like the genus Nephronaias will be included in this new tribe that they have called Papaniidini. However, due to a lack of molecular sequence data available for this particular genus, they were not able to confirm this. So we were able to sequence 3 separate genes and perform a phylogenetic analysis with our specimens of Nephronaias tempisquensis. And we used taxa that were actually directly from that Pfeiffer et al. Study. And from the resulting tree, we were able to confirm that Nephronaias tempisquensis is indeed a part of this new Papaniidini subfamily. And this is supported by pretty high posterior probability and bootstrap confidence values. So just to circle back to the barriers that I talked about before, we were able to confirm that an identification of Nephronaias tempisquensis in Costa Rica. We confirmed the membership of this species in the new Papaniidini tribe. And we are also able to contribute novel molecular sequence data for the genus Nephronaias, which will be extremely useful in future accurate species identification and analyses of phylogeny in this group. So truly a step in the right direction for improving our knowledge on these super interesting creatures. So I would like to acknowledge the following individuals for their support of myself and this project. And thank you all so much for listening and for allowing me the opportunity to share my research with you all.
So our first speaker today will be Hannah Freeman, who will be talking to us about flycatchers.
So just this space for you. Yeah, sure. Hello, everyone. My name is Hannah Freeman, and today I'm excited to talk a little bit about my honors research on determining the effectiveness of forestry beneficial management practices for the olive-sided flycatcher, which is a species at risk bird in Nova Scotia. So we all know that biodiversity loss is one of the most significant environmental issues we face today. Due to this, conservation for species at risk is the focus of much ecological research. Sorry. The olive-sided flycatcher is a migratory land bird and is one of 89 species at risk birds in Canada. It is listed as special concern federally on the Species at Risk Act and is classed as threatened provincially in Nova Scotia. The species breeds in forests across Canada and is threatened primarily from habitat loss. In Nova Scotia, the main driver to this habitat loss is from forest harvesting. And although the olive-sided flycatcher has been found to inhabit protected areas such as national parks, past research has found that these areas are going to be insufficient in conserving this species, and thus we need to apply conservation efforts in working forests. However, this is challenging as we need to balance the interests of both the forest harvesting industry in Nova Scotia along with ecological conservation. One approach to this issue is beneficial management practices, or BMPs. BMPs are guidelines that aim to decrease the environmental harms associated with the process. These are often created and implemented in terms of species at risk conservation. A species-specific BMP was developed for the olive-sided flycatcher in context of forest harvesting in Nova Scotia and was recently applied in a harvest block in Cape Breton in 2023. Some of these recommendations in the BMPs involve specific retention patterns around areas where the species was observed, as well as the maintenance of tall dead trees called snags, which the birds like to feed from. However, before we can apply these BMPs elsewhere in the province and even in their breeding range, we need to determine if they are effective in conserving the species. which is where my research comes in. My study involves the assessment of audio recordings obtained using autonomous recording units deployed within the harvest block. C. Past research has found that the Olivesided flycatcher has song rates that will vary with their breeding status throughout the season. If the Olivesided flycatcher is breeding successfully, they will display decreasing song rates starting earliest in the season to decreasing at the lowest later on. We will be analyzing 40 minutes of recordings a day obtained from May to August in the harvest block. By listening to the songs and viewing the respective spectrograms, we will be able to total the number of male songs as well as pips in each recording. Once complete, we will be able to view the results and compare them to the expected pattern indicative of respective breeding. If we find that the pattern aligns with what we expect of successful reproduction, then we have evidence to support the success of these BMPs. And if we find that they do not, then we know that we may need to make some alterations in order to better preserve habitat for the species. Either way, it's a step in the right direction for Olivesided flycatcher conservation, not only in the province, but across their breeding range. Thank you for listening, and I look forward to taking questions later.
Thank you. Our next speaker is Lauren, who will be talking about radiocarbon.
Hi everyone, my name is Lauren and I'm an undergrad student at Dal. Today I'll be presenting the project I've been working on titled Reconstructing Changes in the Northwest Atlantic Radiocarbon Depth Gradient using deep water Gorgonian corals. So that's a bit of a mouthful, but essentially, what can corals tell us about ocean warming? So we'll start with the Northwest Atlantic Depth Gradient. So this map here shows the Northwest Atlantic Ocean. This is the region of ocean warming fastest of anywhere globally. For this project, we're focusing in on the Scotian Shelf, which is the region just off the coast of Eastern Canada. The mechanisms of warming here are mainly due to the inland push of Gulf Stream water that carries warm, salty water northward near the surface, as you can see in the depth profile here. It's pushing inland relative to Labrador slope water that carries cold, fresh water southward. So we're seeing a greater influence of Gulf Stream water here and also more mixing between these water masses. So now we'll talk about radiocarbon. Essentially, radiocarbon is produced in the atmosphere when cosmic rays interact with nitrogen. Eventually, it's oxidized and turns into CO2. And the CO2 gets into our oceans. Depending on where a water mass sits in the water column, it'll have a different radiocarbon fingerprint. So Gulf Stream water that sits closer to the surface will have a more enriched radiocarbon signature, while Labrador slope water that is more sheltered will have a more depleted radiocarbon signature. Now, it's very important to note that nuclear bomb testing in the 1950s dumped a bunch of artificial radiocarbon into our atmosphere and into our ocean. But this is really useful for us. So if we're looking at the chemistry of marine materials to understand the chemistry of water, and specifically radiocarbon, and we see a large spike in values, we know that this is definitely 1950, which can help us constrain ages and also understand the timing of events. So this is a study of surface water radiocarbon that we produced in our study here. And you can see this really large spike in 1950. So it's definitely something that's very evident and easy to trace. So now I'll talk about deep water Gorgonian corals. So these corals grow on the Scotian Shelf, as shown here, and this is what their skeleton looks like. They have a two-part skeleton of calcite and Gorgonian. So this is a Gorgonian fraction shown here, and they produce annual growth rings, kind of like a tree. We can trace the radiocarbon in these growth rings to understand surface water radiocarbon. If we drill out along the growth bands of the calcite fraction, we can understand deep water radiocarbon. And from this, we can produce really nice depth gradients because we have that calcite fraction showing deep water radiocarbon and the surface water showing, or the Gorgonian fraction showing surface water radiocarbon. And this can help us resolve some problems around changing water masses and warming in the Northwest Atlantic. And that's all, thanks.
Now for our next speaker, Shubhangi, who will be talking about AI and dairy cows.
Hello everyone, my name is Shubhangi. I'm doing my master's in computer science at Dalhousie University.
and today I'm going to talk about innovative AI-based biometric recognition for dairy cows. So when I was a child, I always wonder like how farmers used to manage all of their cows. It could be difficult, right? So when I grow older, I understood it's actually a hard task, especially when they have number of cows, taking care of their milk production, herd management, disease control. It could be problematic. Earlier, farmers relied on methods like tattoo, ear notching, and hot iron branding to identify their cattle. These methods, while traditional, brought pain to the cattle and went foolproof. In places like Alberta, branding is used on almost 20% of the beef cattle, but it's banned across Europe. So finding and searching for a better way lead us to a crossroad between farming and artificial intelligence. Nowadays, we are using a fancy technology called artificial intelligence to help out the farmers. So here what happens is like computers are trying to recognize the cows, just like your phone recognizes your face to open it. So it's the same technology. And as with the advancement in AI, we can give a model data set which it can get more precise and efficient with the time. Instead of getting a physical tag, they get a digital tag, which can be further used for the information about the systems and their breeding time and everything. We can give a non-invasive real-time identification, but no journey is complete without its challenges. So usually cows doesn't face the camera. And the varying lighting and weather condition obstruction makes the picture complicated. So we can give a AI model a data set which consists of several or thousands of cattle images, and it can get more precise and it can get more efficient. We are coming on to the data sets. The data sets will be collected mainly from the open repositories, like so that we can get different breeds of the cows and which we can examine all of them. And we'll also going to the Agriculture University of Dalhousie University, Agriculture campus, where we'll examine around 60 cows and take pictures and videos of them, and especially for this purpose. It's not about just building a technology or just creating something. It's about making a decision support for the farmers. It's about turning a farm into a nurturing community where each cow is taken care for, is understood and known. Thank you.
Next, we have Crystal, who will be talking about security in mobile games.
Hi. Yeah. Sorry, I was just installing a game on my phone. You consider threat of malware when you install games on your phone? Many people don't, What is malware and how can it sneak into seemingly harmless games? Well, malware, short for malicious software, is software that can take harmful actions against us and be hidden on our devices. It can compromise our phone security and steal our information. Many people don't take this threat seriously because they're not thinking about security. They just want a fun game to play. But it's a real and present danger. App stores like Google Play try to mitigate this risk with solutions like Google Play Protect, but they're not always fireproof. For example, in 2021, it was found that Google Play Protect detected only 31% of stalkerware attacks. And that's bad because stalkerware can secretly track and record your phone. Do you like to be spied on? I know I don't. And games, they're not exempt. Just last year, there were found 38 Minecraft clones with hidden adware, and that affected 35 million users. My honors research goal was to analyze Android mobile games for the possible presence of malware using publicly available security tools. I employed VirusTotal, an automated system combining over 70 malware detection systems, as well as 3 consumer tools that I use to test Android games directly on a device. My data set included both known malware and benign apps, as well as 51 games from the Google Play Store. My analysis revealed from the virus total tool set 10 reliable tools with over 90% accuracy, as well as 9 less reliable tools returning type unsupported over 70% of the time. I also found 8 games with suspicious behaviors, suspicious being defined as being marked malicious or having many tools time out or return type unsupported. The game that I thought most likely to be a true positive was Minecraft 2, which was found by Kaspersky to contain adware. A Minecraft clone with adware. Sounds familiar. Maybe it was missed when the other Minecraft clones were found. This highlights the ongoing battle against malware in mobile games and the ongoing need for advancements in cybersecurity, especially in the gaming world where millions can be impacted. My research reveals gaps in the current malware detection systems, even in popular app stores. As technology advances, malware keeps getting more sophisticated. So we need continued research to test and update the games that detect these threats. It's about keeping our digital space safe so we enjoy our favorite games worry-free. Thank you.
Hi everyone, so I'm an undergraduate student at Dalhousie University and my honors thesis is focusing on the impact of precipitation phase on changing groundwater recharge in mountain regions across Canada and US, supervised by Professor Warren Somers. So just to tell you before we dive in, precipitation phase is just how precipitation falls, whether rain or snow, and recharge is basically how much water is being inputted as a groundwater resource. So why do we study this? Well, where do our freshwater resources come from? Well, out of Earth's water resources, only 3% is freshwater, and out of that, 68% is stored in ice and glaciers, and 30% is stored in groundwater. Now, due to climate change, it affects various regions differently. So research has found in low elevation areas, the increase in temperature is about 0.2 degrees per decade, while in high elevation areas like mountain regions, it's around 0.3 degrees per decade. Now, this impacts negatively these mountain regions by firstly, glacier declines, declining late summer snowmelt, changing precipitation phase, and rising surface water temperatures. Now, with this study, it is important to understand the long-term trends of groundwater recharge across various geographical and political boundaries. This made me come up with 3 research questions that generally address how snowmelt or precipitation dominate groundwater recharge across these geographical regions, which is Canada and US. Are snow-dominated wells changing differently than rain-dominated wells? And whether a shift towards less snow and more rain as precipitation will affect the groundwater recharge due to climate change. This is just a study area to show you kind of geographically where the wells are located and where I'll be getting the data from. So you can see the spots are the recharge wells. Now, this is just an overview of my methods. A really big summary of my methods. So the data was collected from various geological data sets. It was filtered based on various criteria. This gave me 171 wells. It was categorized whether it's snow or rain dominated. I'll produce various scatter plots, do some statistical analysis, and then do a much more detailed analysis on the number of wells. And here we can see just on the right side kind of what the wells look like from where the data was not gathered, but just an example. Now, this is one of my preliminary results, just to show you. This is a scatter plot graph that you can see on the y-axis, it's groundwater levels. On the x-axis, it's the month. And each scatter line is the year. So you can see the blue ones are the oldest years and the red ones are the newest years. And this just shows how groundwater is changing over time. Now, why is this important? Well, this study is important to understand how groundwater supply changes over time. Specifically, this will inform various government officials and decision makers of how we can manage our waters in the future if the groundwater levels are indeed changing negatively over time. And it will overall enhance our future hydrological research and contribute to other studies. Thank you. Any questions, which I will receive later.
Hi, everyone. My name is Sarah, and I'm doing my master's research in Dr. Ann Diel's lab here at St. Mary's University. And I'm here to tell you that freshwater and saltwater are vastly different environments. In fact, for fish, this difference is actually a matter of life and death. 97% of Earth's fish species can only survive in either freshwater or in saltwater. But a remarkable 3% of fish species are euryhaline, meaning they can live in both fresh and saltwater. Sticklebacks are a family of fishes with many species who are euryhaline and some species who live only in saltwater. But intriguingly, a few of these saltwater exclusive species can actually tolerate fresh water, which begs the question, if these species can survive in freshwater, then why don't they live in freshwater? And the answer may be that they're not actually capable of tolerating fresh water during their entire life cycle. Our saltwater exclusive stickleback species might not be able to tolerate fresh water during sensitive early life stages. And so I studied baby fish still in their eggs from the 3-spine and 4-spine stickleback who are euryhaline and the black-spotted and white stickleback who live only in salt water. Now, if a species can't reproduce in freshwater, they're not going to persist very well in freshwater habitats. And so the first thing I studied was fertilization success. On this graph here, you can see our saltwater exclusive species on your left and our euryhaline species on your right. And immediately what we saw is that the black-spotted stickleback's fertilization success dropped by 75% in freshwater. This is significant and may actually be the reason that this species does not inhabit fresh water. Our other species had similar fertilization success in fresh and salt water. And what's more is that in all 4 species, once the embryos were fertilized, pretty much 100% of them survived to hatch, which tells us that all babies from all these species appear to do just fine in freshwater. But what is the cost of their survival? If coping with freshwater is a significant challenge for a species, then that species is not going to choose to inhabit freshwater habitats. To see if this was the case for our stickleback babies, I measured metabolism to quantify how much energy it costs them to stay alive. But it turned out that all 4 species had similar metabolisms in fresh and saltwater, and no species appeared to be struggling to maintain survival in fresh water. So in the end, my research indicated that reproduction potential limits species distribution into freshwater, but early life freshwater tolerance does not. This study has helped us gain a deeper understanding into stickleback's unique biology, and I'm currently exploring further to see if the ability to overwinter in freshwater as adults might further limit species distribution. Thank you all very much for listening to my talk, and I look forward to answering any questions.